It's a pleasure to introduce Dr. Mohammed Naveed, who is a consultant pediatrician and consultant geneticist and principal investigator of the first project of CAGS. He is presently Director of Operation at His Highness Sheikh Sultan bin Khalifa Al Nahyan Humanitarian and Scientific Foundation. And he's going to talk about today about search for genetic modifiers of disease severity in Tibial, Hemimelia in large multi-generational Arab family, progress and future directions. Dr. Naveed.
Yeah. Good morning. Thank you, Dr. Kamal. Thank you, Dr. Mansoor. What I'm going to present in the next half an hour is an ongoing research project of Center for Arab Genomic Studies. It's a rare disorder, not a common disorder in most parts of the world. It's called as Tibial Hemimelia. So I'm going to present the status and what we are planning to do next. Now, for those of you who are not familiar with this phenotype, we are talking about one of the long bones of the lower limb that is called as tibia. Now, this condition, hemimelia, refers to either hyperplasia or aplasia of this tibia. If you see on the right-hand side, there are two long bones fully developed. On the left-hand side, the tibia is short and hyperplastic. And consequent to this, it is very common to see other associated malformations that could be the malformations of the lower part of the limb, or it could be other abnormalities. Now, the story begins about 10 years back with this child, who presented to us with an abnormality of the right lower limb. If you look at this left lower limb here, it is perfectly normal. There are two long bones here, but on the right-hand side, there is only one bone, long bone. Now, the family comes out with an information that this is not the only case in the family, There is one more affected, and again, it is the right lower limb. A week later, the father comes out with this photograph of a newborn baby born with same problem, but here there is something additional. There is also something on the upper limbs here. So this looks like that we are looking at a skeletal dysplasia, which may not be common in this part of the world. Then we went further to look, how many more people that could be there in this family. In our first analysis, we discovered that there are about 9 members in this family. We presented this family to the Executive Committee of Center for Arab Genomic Studies, and we convinced them that we should do something. We should do some research here that could lead to an answer to the problems this family is facing. Now, we formed our own group here, and we decided to proceed with the help of a US collaborator who was interested in the developmental limb anomalies. Now, what I'm going to present is what we have done so far with the involvement of 4 centers, the University of Geneva, the Oklahoma, and the Omaha group. Now, we found out that after examining nearly 145 individuals in this large family, 6th-generation family, there were 10 consanguineous marriages, 23 affected, slight male predominance, around 14 males and 9 females. Now, what did we find here? Something very, very interesting, Most of the times, they had abnormality on one of the lower limbs, and almost all cases, it was on the right side. So this made us more interesting. It could be an incidental phenomenon also. In addition to this, many members of the family, they also had what we call it as a reduction defect or acrodactyly of the upper limbs. So it is not just tibial hemimelia, it's a tibial hemimelia associated with other malformation. Now, as we saw more and more cases, we discovered that this tibial hemimelia is not unilateral. So we also had some families in which both the upper limbs were affected. There are also some other minor malformations here. If you can see this, syndactyly, camptodactyly, and other oligodactyly. These were the other associated phenotypes in these large family. These are some of the radiographs to showing some of the cases had complete aplasia of this one of the limbs here. Some of them had hypoplasia and other abnormalities and various degrees of acrodactyly. Now, the first question we asked is, are we looking at something that is already talked about, published, known about it? So we did a literature review, and we decided that we should look at conditions in which tibial hemimelia or acrodactyly is associated. After a careful review of, again, the phenotype and also the literature, we decided that we should look at 4 important regions, 2 on chromosome number 7, 1 on chromosome number 8, and the other one on chromosome number 10. So we selected 18 markers and we tried to do the linkage analysis. What did we find? We found that our phenotype was not linked to any one of these 4 regions we tried to look. So this became more interesting. Maybe we are looking at something new, something different. We tried to present our work in several conferences just to gather more information, and finally we decided to proceed with the publication. So this was our first publication of CAGs in 2006 in American Journal of Medical Genetics. Now, we are talking about the year 2004, 2005. That time the technology was still evolving. Now we have many approaches to gene discovery. I'm just going to present what we could do in those days before the start of the high throughput sequencing and other information that was available. Now again, we went back to the Executive Committee of Center for Arab Genomic Studies, and we said we have to proceed further. In 2006, we thought we should do a whole genome scan. That is the only way to proceed further. So we decided to do genome-wide linkage scan, and we didn't have the facility in UAE, so we did it in, we took the samples to US and we analyzed there. And what did we find? We find an interesting information here. There were 2 loci, one on chromosome number 1, other one on chromosome number 6, which were giving a very close information that we are looking at these 2 loci here. So we tried to see whether all the family members or most of the family members, this data could be reproduced, and we could reproduce the data in many members of the family. And we published these findings in American Journal of Human Genetics in 2007. Now, we proposed at this time, since we had looked at 2 loci, we proposed a hypothesis called as the digenic inheritance, which was well known at the time. And we got the listing, in OMIM. So our family was, localized just to the new OMIM number. Now, soon after these publications, many people joined together. They came out with the information, they communicated to others, say that we also have similar families. And most of them, they contributed not only the clinical information, but also the DNA samples, for a collaborative research. Now, at the end of this journey, with a lot of people involved now, so we were trying to see what to do next. We could have gone again, through the genome-wide association studies. Now we had multiple data. We could have done meta-analysis. We could have done fine mapping, expression studies, or epigenetic analysis. But that time it was very clear that, there are certain drawbacks to this GWAS technique because it is going to fail to identify the rare variants. we may not understand about the structural variants, especially the copy number variants. Okay, gene-to-gene interaction may be more difficult with the GWAS techniques, and gene-environmental interaction is something. So at that time, it was very popular, what we call it as a missing heritability, or the dark matter that cannot be looked at by doing GWAS studies. So the alternative at that time in 2007, 2008 was, okay, the new high throughput sequencing that was available, but it was still very costly. Now this technique is becoming more and more affordable because the cost is reducing, and it has been proven that to discover many novel mutations in these regions. And yesterday Professor Cooper was talking about the availability of these human genome databases, mutation databases. With this information, definitely we can make more meaningful information out of it. Now, we did try to do the exome sequencing, okay, for these two important major loci, okay. And what did we find? We found mutations in FMN2 gene mutation. It's a gene that is involved in cell migration, in morphogenesis, and Philippon gene mutations, okay, in which this gene is involved in cell migration. Now, the other way to look at is the animal models, okay, to look out for the natural variants, the knockouts, and expression. A group in Germany, okay, they came out with some very interesting information, okay. One of the family they had was, even though it's a small family, it was very interesting that it looked like that it is an X-linked inheritance. Now, Germany is a favorite destination for the patients from UAE because they usually go to Germany for reconstructive surgeries, especially for orthopedic consultation. So it turned out to be that, okay, it is the same branch of the family, they were trying to look at it, okay. Now, this group, okay, they found out that there were duplications in chromosome number 17. If you remember this, the previous figure, so we talked about two major loci here. We also had chromosome number 17 that is involved, but it was not significant in our family. And these findings were published by this group. And if you look at carefully here, Anna Rajab is also involved. She's the next speaker here. She's from Oman. And the same family, we have it, the branch of the family is Oman, and these are the same sample that were looked at it. So it looks like that, okay, we are looking at something different. Another interesting finding they talked about in this publication is that the mouse embryos, okay, if there is a defect of the central apical epidermal ridge, this can lead to this phenotype. They also worked on the zebrafish embryos. They found out that this gene that is duplicated, okay, that is VHLHA9, okay, so it is a transcription factor, and knocking out that gene, okay, so the pectoral fin of this fish does not develop. If you look at in this, the wild type here, you can see both the fins here, here and here, but in the knockout mice, you don't see the development of these fins. Okay, this is a very interesting information that is there. So just summarizing, okay, the findings we had 6 loci, of which 2 we considered is major because it was statistically significant. So we find mutations in these 2 genes. The group from Germany, they contributed this information, okay, by showing the duplications of this gene. But still we are working on these genes. Okay. So it seems that we have tried to reach to some small conclusion by following all the 3 major methods by which we can look at the genes which are involved in these complex disorders. Now comes the question, what next? It has become more and more clear as we see more and more cases in this family, as we study more, as we review the literature more. It looks like that we are dealing with a very complex phenotype. So we have to look at susceptibility alleles. There may be protective alleles also. We need to see the relative the contribution of both susceptible and protective alleles. We have to look more into the rare and structural variants in these genes we are going to discover. We have to look at gene-to-gene interaction and gene-environmental interactions. Now I'm going to spend a minute on this gene-environment interaction because this is something that I personally feel we should look at it. Now, one of the phenotype in our family is the acrodactyly, which I showed you about. If you look at the number of people who are affected with acrodactyly, in addition to tibial hemimelia, is nearly 20 of these people have this acrodactyly. Now, this acrodactyly is not only described in humans, it is also described in chicken, frogs, and mice. Okay? And it has been shown that the pregnant rodents, when they are exposed to either retinoic acid or cadmium, they develop acrodactyly. So it is possible that an environment also could be contributing to this phenotype. Now, if there is a relation, if there is an interaction, then we have to understand what exactly is this, the correlation of the environment with the phenotype that is evolving in our family. So let me just briefly explain that maybe as a next direction, we have to look more into this correlation, whether it's a passive correlation, whether it is an active correlation, or it is a reactive correlation. Just to explain to you, I just took this example here. Let's say, for example, if you're looking at a musically talented kid, one of the things that could ask it is something that is running in the family, or it's something that the child has acquired. Now, if you look at this particular family here, a large family with many members, many musicians, so it is possible that, this trait may be running in the family. Now, what is a passive correlation? Suppose if this is the kid born to a family in which there are already talented parents, so it is possible this child is growing by hearing the music right from this. At the same time, he has an access to these instruments. So it is a passive contribution of the environment, for the development of that particular trait. Now, when do we say it as active? For example, if these kids were also exposed to musical education, we are talking about an active contribution of the environment. And what is reactive? Suppose if these kids also undergo special education, special training, special supervision, so we can say the environment is also reactive. So it is possible that we may have to look into these lines if you are looking at the gene-environmental interactions. Now, as the time goes, we are, something that is going to happen is the advances in the technologies, whether it is GWAS or whether it is, next generation sequencing or it is in biome, it is bound to happen. But what is more challenging is, the work the people have to do in the background. The people who are involved, who are looking at the clinical phenotype, they need to look at more and more cases. They have to follow up the already existing cases. They may have to follow the new cases that are occurring in this family. And this information should be passed on to this lab, and the lab should do more and more work. And the bioinformatics team, should be interacting on a regular basis. And when this happens, we have success here. Now, to have this success, what is important is to have a new breed of scientists who understand the anxiety of the patients who are waiting for something to happen, the problems in the lab, and the amount of data that is coming out with the new technology, the interpretation of this. So unless, we have a very good network of scientists involved in this in this phenotype, so it is going to be a long way. I'm going to end my discussion with this slide. At the end of the day, okay, we are to help these patients with complex disorders. We have a moral obligations as clinicians, as the people working in the lab, and also as a community, that we have to find some solution to these patients. But it is very important to understand that it's a long way before we help these patients. Okay, so to this we find one more obstacle that we commonly see in most parts of the world, that it's not only to find clinical geneticists who are involved or who are interested in such phenotypes, okay, who can give more time, okay, more effort, okay, to define it better. The scientists who are who are involved in such type of activities, especially those who are concentrating on the developmental genes, the team of bioinformatics, okay, and lastly, the fourth piece of puzzle is the decision makers who decide the future of this research. So without funding, there is no research, and without research, there is no invention, and we will not be able to offer diagnostic to these patients. So it seems that, okay, there are many issues that delays the research, because yesterday somebody was asking, Dr. Naveed, still you are working on the same phenotype for almost 10 years. So we have our own problems here. So it's possible that these problems are there. Unless these problems are sorted out, I don't think we can give an answer to these patients, okay, in a short period of time. I think I'll end my discussion. Okay, if you have any questions, I'll be happy to answer.
Thank you very much, Dr. Naveed. Since we have time, actually, we are happy to take a couple of questions as well. We are in good time. Just switch the microphone on, please.
Is it on? Hi, great talk. I'm Juan Rodriguez from Cornell. I have a question in terms of, so at the end of the study, you have at least 4, maybe more variants that are linked to this phenotype.
Yeah.
But then later you found that there was some environmental factor that could be causing it.
Yeah.
Did you go back into these pedigrees and then look at those environmental factors?
It is, this is an Emirati family. Okay, we had a very tough time. Even okay, these are some of the problems most of the clinicians face, especially in this part of the world. Okay, and we had a very tough time, okay, bringing these patients to the clinic for evaluations. In fact, most of our work was done in their homes, okay, and all the assessment, especially for the females, we had to do it at their places. But some of them we could manage to do, bring them to these the clinics and to the hospitals for radiographs and other investigations. So, but this is something that we presume that we should look into the environmental contribution to this phenotype. But I really don't know, to be honest, that how to proceed further. I look forward for some more collaborators who can throw more light on this way to look at it. But it's my presumption that ectrodactyly seems to be one important phenotype that may be influenced by the environment.
Okay, and then in terms of the total number of variants that you have now that are linked to this phenotype?
Yeah, even though we found when we first published in 2007, there were almost 6 loci here. We consider 2 as the major one. That's how we proposed a theory of digenic inheritance. But it looks like that we are looking at an oligogenic phenotype. So there may be more and more genes involved. But as we see more and more cases, I think maybe it may turn out to be a more complex phenotype.
Yes.
Yes, please.
There's a microphone behind you. If you use it, everybody could hear the question. Thank you.
Can you hear me now?
Yeah.
Dr. Mohammed, thank you very much for this excellent presentation and excellent work. Now, you said that contributors from different parts of the world, they shared the family's DNA, right?
Yes.
Okay. Now, your family is from the UAE. Do they have a distinct phenotype that you can correlate with the findings, the two mutations you identified?
No, we have not.
And sorry, doctor.
Yeah.
Have you done any functional experiment on those two mutations?
No, not yet. It's just new information, okay, that we have not yet published actually. So this is the information that I've just revealed here. So we are in the process of, trying to work on other families that we have from other contributors, okay. What I'm presenting is what we found in our families. It doesn't mean that we'll find in other families, but we have access to those samples and we are working more on that.
So you are looking for a distinct phenotype that will distinguish your families from the other contributors, right?
Yes. The phenotype seems to be almost relatively more homogeneous, what we find in our family and other family members.
Right.
Okay. But we are not sure that what we are finding in our families, we can reproduce it in other families. Unless we do that work, we will not be knowing it.
Right, doctor. Thank you very much.
Thank you very much, Dr.
Thank you.